Okay, I think we might start because I will warble on for a little bit. So, welcome everybody to this Wednesday meeting, and this is the first in a series of 4, which I am delighted to introduce. So, for those of you that don't know me, my name is Andy Jennery. I have a number of roles and hats. I'm a paediatric immunologist working on the transplant ward, but late last year, I was appointed as the James Spence Professor of Child Health. And my goal is to increase awareness and work around academic paediatrics in Newcastle, in the hospital, and hopefully eventually around the northeast and the greater northern region as well. And as part of that, I thought an interesting thing to do, first of all, would be to introduce you to some of the new work that is being undertaken by some of our rising stars. Now, if you are following an academic training pathway, the final position that you may get before you become a consultant is known as an academic clinical lecturer, or an ACL, and these are NIHR funded posts. Now, in the UK, I think predominantly actually in England, there are an average of about 3 to 4 ACLs awarded in paediatrics every year. We were extremely lucky last year because we had 4 paediatric ACLs appointed here in Newcastle. And I have asked each of them over the next few weeks to introduce themselves to you and tell them a little bit about what they're doing, maybe what they did for their PhD and their plans for the future. So it's with great pleasure today that I welcome Iram Haq, who is our pediatric respiratory ACL, and she's going to tell us something about what she's been involved with. Thanks, Iram.
Thanks, Andy. So I will just share my screen and hopefully you'll be able to see my talk. So can everyone see that okay?
Yeah, perfect.
Perfect. Okay, so my name's Iram Haq, and thanks, Andy, for that introduction. So I'm going to talk to you a little bit today about some of the research that I've been doing in cystic fibrosis. So some of you will have heard some of this before, so apologies for that, but hopefully there is some new material in there as well. So I'll talk a little bit about my research experience so far. I'll mention obviously cystic fibrosis and the significance of the airway surface in CF. I'll talk to you about the aspects of my PhD research and also my sort of current and future research directions as well. So I first got a flavour of research when I did an intercalated MRes with Andy Jennery, and that was way back when I was in medical school, and then sort of embarked on my foundation in paediatric training, and then did a PhD as an out-of-programme sort of research post, which was funded by the Wellcome Trust. Started my respiratory training in 2018, and then was awarded an NIHR last year to continue my research interests. And at the moment, I'm based at Newcastle in the university and part of the Children's Research Group. So this is led by Malcolm Broadley, which many of you will know, and other sort of key members in the group are Aaron Gardner, who's a postdoc, Noreen Zainal Abidin, who many of you will know, and Alexandra Leberka, who's also supervised by Andy Jennery, but also carries out some of her work in the same group. So cystic fibrosis is an autosomal recessive life-limiting disease. It's caused by defects in the CFTR protein, and this is a channel which transports chloride and bicarbonate ions into the surface of epithelia where it's expressed. And so CFTR is present in lots of different epithelia in the body, and this gives rise to the multi-system nature of the disease that we see in CF. And obviously I'm interested in respiratory medicine, but we know that by far the mainstay of morbidity and mortality lies within the respiratory tract. And as I say, that the disease is life-limiting. So just to kind of put this into context, I'm just going to talk a little bit about one of our patients that we look after here in Newcastle who has cystic fibrosis. So he's an 8-year-old boy. He was born with his birth weight at term less than a 0.4 centile. and had a difficult postnatal period with an ileal volvulus and atresia, requiring a laparotomy and ileal resection. And subsequent genetic testing as a result of this postnatal kind of clinical course led to a confirmed diagnosis of CF. And he was found to be homozygous for the most common mutation that we see in CF, which is F508del. And this is a severe disease-causing mutation. And he had a sweat chloride, which is also another part of the diagnostic workup, which showed an elevated sweat chloride of 103. So anything greater than 60 is abnormal. So in terms of his progress, he isolated Pseudomonas in 2016 and 2018, and we know that Pseudomonas is quite significantly relevant. It has significance in CF in terms of causing respiratory disease, and also isolated something called Mycobacterium avium complex, so a non-tuberculous mycobacterium in 2018. He had a CT scan at that point, which showed features of bronchiectasis in his right upper and left lower lobes. And as part of routine surveillance, when he had an ultrasound of his abdomen, he was found to have early signs of CF liver disease, namely some features of fibrosis, and also an elevated prothrombin time on his bloods as well. And his ongoing management to date essentially consists of fairly standard sort of medications that you may expect. So he's on daily mucolytics, he's carrying on his physiotherapy, he's on pancreatic enzyme supplements or Creon, he's on fat-soluble vitamins, and ursodeoxycholic acid, which was started last year in conjunction with the diagnosis of the early CF liver disease. But nutritionally, he's doing quite well. His weight and his height are on the 50th and the 75th centiles, and his lung function has been relatively stable at around 80 to 90% predicted. And I should add that he started treatment with Orkambi at the beginning of this year. And Orkambi is one of these CFTR modulators, which are relatively novel therapies in CF, and they've really sort of revolutionized therapeutic strategies in CF disease. So these new therapies have been developed based on an understanding of what happens to the CFTR protein in terms of how it's made within the cell, and then how it mobilizes up to the surface of the cell where it functions as a CFTR channel. So they can be broadly classified into two groups of medicines. You can get correctors, which help mobilize the faulty CFTR towards a plasma membrane, and then potentiators, which can help the dysfunctional CFTR work as a channel when it gets to the membrane. So there's been a few modulators that have been developed and accessible over time, and most importantly, triple therapy was a really exciting announcement for 2020 because of the significant improvements that have been shown with lung function and other clinical outcomes in people with CF in the clinical trials where it was investigated. And these therapies in combination will apply to and benefit around 90% of people with CF. But there's actually still a smaller group, so around 10% of people with CF who will still need a form of treatment that will help provide some comparable clinical outcomes to those modulators that I've talked about just now. So in order to help understand how we may be able to help some of these people, it's important to understand about the airway surface in CF and how it's abnormal. So if you imagine these are the cells that line the surface of the airways, so the airway epithelial cells, and above those we have the cilia, this layer here called the airway surface liquid, which helps to clear mucus and prevent the build-up of infection and inflammation within the airway. But in cystic fibrosis, because CFTR is dysfunctional, we don't get chloride and bicarbonate transport into the airway surface. And the overall effect of this is to create a thick and acidic and dehydrated airway surface, which is hostile. It leads to defective mucociliary clearance and ongoing infection and inflammation, which are the hallmarks of respiratory disease in CF. But there are other channels that sit in the airway alongside CFTR. So there's this channel here, which is the epithelial sodium channel, which is really important. And I'll talk a little bit about that later on. But this was the channel that I was interested in my PhD, which is another chloride channel that also transports chloride and bicarbonate like CFTR. And so therefore it's possible that by activating this channel, you could compensate for some of the problems that we see with abnormal CFTR and help restore some of the airway surface homeostasis that is abnormal in CF. So how can we investigate these channels in the paediatric airway? Well, we need relevant models in order to be able to do this. And we know that airway epithelial cells, so cells that have come from the lining of the airway surface, have been really instrumental in terms of developing CFTR modulators to date, specifically ivacaftor, which was first kind of described in airway epithelial cells in 2009. So how can we collect these cells? So the cells can be collected via a number of different methods. So you can have bronchial epithelial cells, so from the lower airways, and these can be harvested by brushing the airways of explanted lung tissue. And fortunately, I should say, we don't have access to pediatric explanted tissue because clearly sort of transplant occurs with a greater frequency in an adult CF population. And also we can collect bronchial epithelial cells during a flexible bronchoscopy by inserting a brush down the bronchoscope and then collecting the cells that line the surface of the airways. But also another way that we can collect the cells is by using nasal cells. So these can come from brushing the inferior turbinate, so the space between the bony ridges inside the nose, using a cytology brush. And actually, we can do this in children when they're awake, and we do this for children for diagnostic purposes in conditions such as primary ciliary dyskinesia. Clearly, if we're doing a flexible bronchoscopy, it requires a general anaesthetic, whereas if we're doing nasal cultures and nasal brushings, it's possible to do these in awake children, and therefore it lends itself quite nicely to a sort of an accessible paediatric model that has quite significant translational benefits. So the second part of my PhD was to look at if we could use nasal cells as a valid experimental model in CF. So during my PhD, we recruited children with and without CF that were attending for a clinically indicated bronchoscopy. And at that time, we collected both nasal and bronchial brushings from children when they were asleep. As you can see here, over that time, all of the children with CF that we approached themselves and their parents consented for us to be able to collect their samples, and quite a good proportion of children without CF also consented to having brushings taken. I guess the key thing to take from this table here is this is a split of the CF and the non-CF children. It's important to know that the non-CF children weren't free of disease. As I've mentioned, the cells were collected during a clinically indicated bronchoscopy, so they were having a bronchoscopy for a clinical reason. And by far the majority of reasons for doing that was to investigate history of recurrent chest infections, but they didn't have a diagnosis of CF. So after collecting the nasal and bronchial brushings in theatre, I took them to the lab, and this is where I spent a lot of the time growing the cells. And so we grew them to something called an air-liquid interface. So you can culture the cells using a specialist medium and expose the cells to the air and encourage them to develop those typical characteristics that you would expect of an airway. And if they were still alive at day 28, you can see I was still quite happy sort of working away in the lab. So I hope these videos work, and if they don't work, it can be a bit risky showing videos sometimes, but I've got some pictures to demonstrate this as well. So this image on the left here is a picture of what a kind of cluster of epithelial cells when you collect a fresh brushing. And when you look at it down the microscope under magnification, you can see the cilia kind of beating around the edges, and that's just quite nice to be able to show that. And then when you culture those at 28 days, if you imagine you're looking down at the surface of the culture, I'm not sure if you can see that, but you can appreciate some flickering as you're looking down, and that's actually all the cilia that are beating in the culture at that time. But if you're not able to see that in the video for whatever reason, I've just got some pictures to demonstrate that the nasal cultures that we grew did have cilia, and they were ciliated, which is really important. So the red stain here stains for a specific cilial marker. And these electron microscopy images just show the kind of ultrastructure of the cilia that you would expect to see. And they also produce mucus as well. And you can see the mucus droplets on this microscopy image, but also some specific stains, and looking for specific mucins in the airway, they're also present. And importantly, because I was going to be looking at ion transport in the airway, it was important to know that the cells that I was growing weren't leaky. So I looked specifically for tight junctions. So these are the proteins that connect the cells together and give the cells that sort of layer of integrity, and that was also present in the cultures. So having demonstrated that the nasal cultures were relevant and were differentiated and, doing all the things that airway epithelial cells should do, we then went on to look at whether or not we could use these cultures to look at those channels that I talked about before. So specifically this alternative chloride channel, TMEM16A. So I did this by using a fairly old technique, but it's quite a good technique for looking at how channels work in epithelia. And this is called an Ussing chamber. And again, this is another room where I spent quite a lot of time doing my PhD. So this is what the Ussing chamber looks like here on the right. And then this is the kind of experimental sort of setup around it in terms of the equipment that you need. And this is a diagram just to demonstrate what it does. Essentially, you can load your epithelial cells into the chamber. And because you can, we're interested in how channels work, you can apply known activators and inhibitors of the channel to see how well the channel functions. And this equipment here can measure that as a readout. So I was specifically interested in CFTR, which we know is abnormal in CF, and this protein TMEM16A, which is this chloride channel that I was interested in. So this is a typical readout that you get from a nasal culture from a child without CF. So if you just kind of ignore this bit here, but focus on this area, this activates CFTR and this inhibits CFTR. Because we see this rise and decline, we know that CFTR function is working well, as you would expect in a child that doesn't have CF. If you remember, I talked about our child at the beginning with CF, and he kindly participated in the research as well. And you can see that when we add these reagents here, nothing happens. And we know here that CFTR isn't working properly. So it's a really nice way of being able to demonstrate how well a channel's working. So I looked at a range of cultures, so nasal and bronchial, from children with and without CF. And the children that had CF had a range of different CFTR mutations, and was able to show that there were no differences between the nasal and bronchial cultures in both groups, which is great because it means that we can use the nasal cells as a representative model, but also that, as you would expect in the CF group, we didn't have any CFTR activation. However, I just draw your attention to this purple dot here, which represents cells that were taken from one specific donor, and I'll just talk a little bit about them at the end too. So I was interested in this other channel, TMEM16A, and using another reagent that activates this channel, I was able to show that you can actually activate it in the nasal cultures of children without CF and with CF. And that's really important because if we're thinking about a sort of therapeutic strategy for people with CF, it's important to know that that channel is actually there and is working. I used another way to show that the channel was expressed in the nasal cultures, and that was using immunofluorescence. So these green markings are, so if you imagine you're looking down at the culture, the individual blue dots are the nuclei of the culture within the cells, and the green markings are where this protein TMEM16A is expressed. And it's actually expressed in very low levels in the airway, and you can use you can apply certain pro-inflammatory cytokines to upregulate the expression of this protein. And this was just a way really of proof of concept of showing that that it was capable of, that we could capably upregulate the expression of this protein. And you can see that it's mainly expressed at the top of the protein at the kind of epithelial surface when you upregulate it. I also looked to see if there were any differences between nasal and bronchial cultures and CF and non-CF in terms of how much you could activate this channel. And actually, there weren't really any significant differences between CF and non-CF. And it's really good to know that actually the differences between the nasal cultures were not apparent. So we can use nasal cultures to investigate this as a surrogate for bronchial cells. And finally, at the kind of final part of my PhD, I looked at whether or not we could apply novel activating compounds to try and upregulate how much this alternative chloride channel works. And again, this graph here, this rise is a rise in chloride secretion by the channel. And you can see that it's quite a sustained response going on for about 30 minutes or so. And this is different to some of the typical molecules that would have been used to activate it in the past. And actually, this was a really nice finding to show that you can activate this channel and you can promote ongoing chloride secretion using this compound. And this does require some further investigation. So in summary, in terms of my PhD findings, I hope I've shown you that nasal cultures can be a feasible way of looking at iron transport in the pediatric CF population, that this alternative chloride channel is expressed in the CF airway, and that there is potential to improve chloride transport in the airway and potentially restore the homeostasis of the airway surface with some of these compounds. But there are limitations to the culture process. So there were significant challenges with setting up and optimizing the cultures. But actually, I think now over time, we've been able to significantly improve the success rate. And actually, these are working quite nicely. There is a limited resource with this type of technique, and that's particularly apparent now, I guess, in the current climate of COVID-19 in terms of accessibility to samples such as this. And there's also quite significant interdonor variability in terms of how channels are functioning. But actually, I think this is a really important finding because it shows that there is a need to adopt a personalised approach for interpreting some of these functional studies that we're able to carry out using the cultures. And just to try and demonstrate this a little bit more, if you remember, I drew your attention to this purple dot before, so these are cells that came from an individual donor with CF, and you can see that they actually have got more chloride transport than you would expect, showing that there is some function of CFTR ongoing in comparison to the other CF donors. And actually these cultures came from a boy that's nine years old. He's a heterozygote, so he's got one of the severe F508del mutations, but he's also got this other mutation called R751L. And actually this mutation is really poorly described in the literature, and there's just a handful of case reports at the moment, so we don't really know how this affects protein function. His sweat chloride was elevated at 74, but if we think back to the case that I presented at the beginning of a child who's got quite characteristic severe disease, his sweat chloride is lower than what we saw in him. He's pancreatic sufficient. His height, he's gaining weight and growing really nicely with his height and weight on the 91st centiles, and his lung function is actually excellent at 116% predicted. He's got quite sort of relatively intermittent and mild respiratory symptoms. He did isolate Pseudomonas in the past, but he's got overall quite a mild disease phenotype. His chest X-ray imaging has been fairly normal, and he hasn't required any CT imaging to investigate for bronchiectasis. So actually there's quite a difference between the two patients, and you can see that from the functional studies that we're able to carry out. So we looked into this child in a bit more detail and actually spoke to our adult CF colleagues, and it's apparent that this child also has an aunt and uncle that are looked after by the adult CF team, who also have this R751L mutation, but another mutation called G551D, which is also associated with severe disease, but is present in a smaller proportion of the population. Their sweat chlorides range from around 71 to 100. They were both pancreatic sufficient, and their BMIs are actually high and within the normal range. They have fairly standard sort of normal lung function, and although have isolated between them Pseudomonas and Haemophilus in the past, actually the disease is quite mild. So we looked back at the results for our individual child who had this mutation, and this is the kind of chloride transport by CFTR. And as I've mentioned already, we can see that this is a normal non-CF child, this is a severe CF disease, and this is our sort of child with R751L, and you can see that there is some chloride transport there. It's not as much as you would see in a non-CF child, but it's definitely more than you would expect in a sort of typical CF case. So we presented this at one of the international CF conferences, and this led to a collaboration with another group to see if we could look into how this protein and how this mutation is behaving. And essentially what we were able to show was that how, in terms of how well the protein is made and in terms of how well the protein is able to get to the cell surface, actually the R751L mutation behaves in a similar way to normal CFTR or wild type CFTR as it's shown here. So this kind of led us to think what's going on with this child. What implications does R751L have in terms of its effects on CFTR? And It's possible, and the further investigation has been really helpful, but it is possible that the phenotype that we see may well be associated with abnormalities in another protein that sits in the airway. So this is this epithelial sodium channel that I mentioned before, which is also a really key player in terms of regulating the airway surface homeostasis under normal conditions. And so we went back and looked at how well this channel, ENaC, functions. So this is non-CF, this is severe CF disease, and the channel functions quite normally. But in our child with our R751L, the channel actually wasn't functioning well at all. So since this work, we've been able to screen this child for ENaC mutations, and the results are still awaited for this. And actually, we know that ENaC mutations are associated with some causes of non-CF bronchiectasis, and this is an avenue that I'm sort of looking to explore further during the course of my ACL, and hopefully I can update you with a bit more about that at a later stage when I've carried out some more work with that. So I just finished with some acknowledgements, and like with all of these things, there's always lots of people that it's really important who have been crucial really to some of the research that's been carried out. But most importantly, the children and parents and the CF and respiratory teams and all the other clinical teams that have been instrumental to being able to collect and sample the airway epithelial cells. I've just put my email addresses down on the bottom in case anyone wants to has any general questions at a later stage or wants to talk to me a little bit more about either some of the research that is going on or any advice about pursuing sort of research in the future. And I'll just welcome any questions. Thank you.
Thank you very much. That was really interesting. And I think really exemplifies what we're very good at in Newcastle, which is this translational research. So it's taking patients who've got problems, trying to take that problem back to the bench and solve it, and then taking that solution back to the patient so that we're actually improving outcomes for our patients. That's a wonderful talk. Thank you. So I'm going to ask if anybody has got any questions. I guess you unmute and ask a question. But I'm going to start with one, if I may, and that is, I thought that cystic fibrosis was now able to be screened for on newborn screening.
Yeah.
Is that correct?
That is correct, yes.
So do you think the sorts of things that you've been talking about will be able to be applied to patients who are picked up very early?
Potentially, yes. So I think the ideal situation, I suppose, with the sort of new modulator therapies that I talked about at the beginning, so the ideal situation would be where you have a child that's picked up by newborn screening and then they're started on, sort of relevant medicines at that time point. And I think that would be the kind of ideal situation. We know that at the moment, those therapies are relatively new. So, even the oldest of those was started around 7 years ago. The triple therapy that we've got has come out now. So it will be interesting to see the longitudinal data in terms of how patients respond to those therapies, what are the overall implications in terms of disease burden, and, are we able to reduce some of the other medications that they may be on? There's lots of things that need to be explored, but the ideal situation would be that you would start a child, as soon as you've got that diagnosis, on whichever sort of relevant therapy is appropriate.
Yeah. And then just another question. Again, and this is completely not my field, but I was under the impression that gene therapy might be being developed for cystic fibrosis. How close is that, and how would that fit with some of the therapies that you've been talking about?
So gene therapy is the ultimate sort of ideal therapy for a condition like cystic fibrosis. But actually it has been a fairly difficult process so far in terms of achieving being able to target the gene therapy appropriately. So there's various different methods that have been used using viral and non-viral vector approaches. But actually there's still a lot of work that's in progress.
Okay.
huge kind of consortium that's involved in that work at the moment, and there's a great deal of efforts that are still ongoing, but still some progress to go.
Okay, all right, thank you. Anybody in the virtual audience got any questions? Oh, Frederick.
Iram, thank you for a very interesting talk, and also I have to say very well delivered. I think that was a very comfortable presentation, so well done. I don't have any prizes, but you would certainly get one for that. So my question is the following. So you've talked about the CFTR receptor and then, of course, the TMEM receptor.
Yeah.
Now, the human body in general is very good in trying to compensate failing mechanisms by overactivating other mechanisms.
Yeah.
And in addition to your efforts to try and stimulate the TMEM receptor in order to overcome the deficits that are there throughout the CFTR protein, is there any evidence that the human body already upregulates TMEM function in CF children?
Yeah.
So by itself?
Yeah. So that's a really good question, and there as with a lot of things in this kind of field of science, there are sort of two schools of thought. one that says that it's upregulated, and one that says it's potentially downregulated. I have to say that and it's a small group of patients that I looked at, but I didn't see any differences in terms of the results that I found. And I don't know whether that's because I looked at a pediatric population that have relatively mild disease, and if there are any changes, whether it's something that develops over time. I think we know that if you knock out TMEM16A in mice, certainly you can develop a sort of CF disease-like phenotype.
Okay.
So we know that we know that it's important for chloride secretion, but then we also know that there are differences in terms of how CFTR is expressed in mice models compared to human models, and the same applies for TMEM16A as well. So I think there is ongoing work at the moment by another group, sort of down in Brighton, who are looking at TMEM16A as a potential therapeutic target certainly in adults with CF.
Okay. Thank you. Following on from that then, so for example, if your serum sodium goes up or down, your body will sense it. So is the body able to sense electrolyte disturbances in lung mucus epithelium?
Yeah, so it can, and I guess that's how you get the kind of abnormalities that you see in CF, because it's to do with the chloride and bicarbonate dysfunction in CFTR that causes those issues. I think that there are lots, and that was a very simplified version of the airway epithelial cell that I showed before, because there are a multitude of channels that sit in the airway. You've got proton pumps, and you've got, sodium bicarbonate exchanges, and there's many, many more, and they are all interlinked, and they all will interact with each other. I think the important thing to take from this is the fact that you can look at individual function from a specific child, and that's where the sort of personalized approach will come in terms of how much does X patient need more of to be able to functionally restore a healthy sort of balance in the airway. But there's definitely that complex interplay.
Cool, very cool.
Okay, any other questions from the audience? Not that I can see. There's a red spot that keeps moving, but I think it's just the recording button. I don't think it means anything other than you're being recorded. Okay, well, I think we've kind of probably reached end of time as well, so I'm going to bring this to a close. So first of all, thank you very much, Iram, for introducing this series for us. And I'm sure if there are members in the audience, and particularly trainees who might be interested in pursuing research, then by all means, please come and see me. But if you want to see someone who's really approachable, then speak to Iram, or indeed to our other three ACLs, who Aisling Flynn, Claire Wood and Sunil Bhopal, who will all be giving talks later on in the series. But what I'd like to do is stimulate some research interest amongst the whole group. I think there's a lot of things on offer in Newcastle, and I think Iram's done a great job in showing off one small part of that. So thank you.
Yeah, and like I said before, I'm happy if anyone wants to email me about anything. That's absolutely fine.
Super. And on that theme, so Andy already said there are 4 ACLs. The next talk next week is by one of the other ACLs. So if your interest is piqued by listening by kind of an academic slant on a clinical talk, then please join us again next Wednesday at the same time.